How does TB bacteria replicate within human macrophages? Imagine tiny bacteria sneaking into our body's immune cells and setting up shop inside them. That's exactly what Mycobacterium tuberculosis does when it causes tuberculosis. These bacteria are clever and have developed ways to survive and multiply inside macrophages, which are immune cells meant to destroy invaders. When TB bacteria enter the body, they are usually swallowed up by macrophages in the lungs or other tissues. Inside these cells, they are trapped in a compartment called the phagosome. Normally, the phagosome would mature and fuse with lysosomes, which are like cellular garbage trucks filled with destructive enzymes and acids meant to kill bacteria. But TB bacteria have evolved to interfere with this process. They release factors that block the maturation of the phagosome and prevent it from acidifying. Without acidification, the bacteria can survive comfortably inside the phagosome. Some TB bacteria even damage the membrane of the phagosome and escape into the cytoplasm, the fluid inside the cell, where they can continue to grow and multiply. The ability of TB bacteria to replicate depends on the state of the macrophage. For example, macrophages that are stimulated by a molecule called interferon gamma, which is part of the immune response, can sometimes become more permissive to bacterial growth. Interestingly, in some cases, this stimulation can lead to the macrophage dying in a way called necrosis, which makes it easier for the bacteria to spread. When macrophages die by necrosis, their cell membranes break down, releasing the bacteria into the surrounding tissue. This creates a perfect environment for TB bacteria to multiply and infect new cells. Macrophages also respond to TB infection by increasing the number of tiny organelles called peroxisomes. These organelles produce reactive oxygen species, which are molecules that help kill bacteria. However, TB bacteria that have escaped into the cytoplasm can be targeted by these reactive molecules, although bacteria inside intact phagosomes are less affected. Another important defense mechanism is autophagy, a process where the cell essentially digests its own components, including bacteria. Proteins like ATG7 and ATG14 are essential for autophagy to work properly. When autophagy functions well, it can help contain and destroy TB bacteria. But if autophagy is impaired, the bacteria can replicate more freely. Overall, TB bacteria have a range of strategies to survive and multiply inside macrophages. They inhibit the normal destruction process, escape into the cell's cytoplasm, exploit cell death pathways like necrosis, and evade immune responses like reactive oxygen species and autophagy. This complex battle inside the macrophage allows TB to persist in the body, making it a challenging disease to treat. Understanding these mechanisms is crucial for developing new therapies that can target the bacteria where they hide and multiply. If you are concerned about tuberculosis or think you might have been exposed, always consult a healthcare professional for proper testing and treatment options.